Coronavirus COVID-19 by Kamal T. and Shubhashish Thakur. Coronavirus COVID-19 Vol. I. Books by Kamal T. Download ebooks, Kindle, for just 50 rupees at Amazon. Download ebooks, PDF, for just 25 rupees at PayPal. Note: links are given below in video's descriptions. Coronavirus COVID-19 official link plus helpline mobile number only for India. Official website https://morph.gov.in. Helpline number 1075 plus 9111 23978046, toll free. Official link for Coronavirus COVID-19 COVID Visualizer. COVID-19 Coronavirus Pandemic. WHO COVID-19 Situation Dashboard. Coronavirus COVID-19 Map. References: Wikipedia, World Health Organization (WHO), Google. Coronavirus.
What is coronavirus? Coronaviruses are a group of related viruses that cause diseases in mammals and birds. In humans, coronaviruses cause respiratory tract infections that can be mild, such as some cases of the common cold, among other possible causes, predominantly rhinoviruses, and others that can be lethal, such as SARS, MERS, and COVID-19. Symptoms in other species vary. In chickens, they cause an upper respiratory tract disease, while in cows and pigs they cause diarrhea. There are yet to be vaccines or antiviral drugs to prevent or treat human coronavirus infections. Coronaviruses constitute the subfamily Orthocoronavirinae, see Figure 1, in the family Coronaviridae, order Nidovirales, and real riboviria. They are enveloped viruses with a positive single-stranded RNA genome and a nucleocapsid of helical symmetry. The genome size of coronaviruses ranges from approximately 27 to 34 kilobases, the largest among known RNA viruses. Orthocoronavirinae. Figure 1. Transmission electron micrograph, TEM, of avian infectious bronchitis virus. The name coronavirus is derived from the Latin corona, meaning crown or halo, which refers to the characteristic appearance reminiscent of a crown or a solar corona around the virions, virus particles, when viewed under two-dimensional transmission electron microscopy, due to the surface covering in club-shaped protein spikes. Figure 2. Illustration of the morphology of coronaviruses. The club-shaped viral spike peplomeres, colored red, create the look of a corona surrounding the virion when observed with an electron microscope. Types. Types of coronavirus. Coronavirus. Alpha coronavirus. Beta coronavirus. Gamma coronavirus. Delta coronavirus. Discovery. Discovery of coronavirus. Human coronaviruses were discovered in the late 1960s. The earliest ones discovered were an infectious bronchitis virus in chickens and two in human patients with the common cold, later named human coronavirus 229E and human coronavirus OC43. Other members of this family have since been identified, including SARS-CoV in 2003, CoV-NL63 in 2004, HKU1 in 2005, MERS-CoV in 2012, and SARS-CoV-2, formerly known as 2019-CoV, in 2019. Most of these have involved serious respiratory tract infections. Etymology. Etymology. The name, coronavirus, is derived from Latin corona, meaning crown, or wreath, itself a borrowing from Greek kappa omicron rhonuita corn, garland, wreath. The name refers to the characteristic appearance of virions, the infective form of the virus, by electron microscopy, which have a fringe of large, bulbous surface projections creating an image reminiscent of a crown or of a solar corona. This morphology is created by the viral spike peglomerase, which are proteins on the surface of the virus. Morphology. Morphology. Figure 3. Cross-sectional model of a coronavirus. Coronaviruses are large pleomorphic spherical particles with bulbous surface projections. The diameter of the virus particles is around 120 nm. The envelope of the virus in electron micrographs appears as a distinct pair of electron-dense shells. The viral envelope consists of a lipid bilayer where the membrane, M, envelope, E, and spike, S, structural proteins are anchored. A subset of coronaviruses also have a shorter spike-like surface protein called hemagglutinin esterase, He. Inside the envelope, there is the nucleocapsid, which is formed from multiple copies of the nucleocapsid, N, protein, which are bound to the positive since single-stranded RNA genome in the continuous beads on a string-type conformation. The lipid bilayer envelope, membrane proteins, and nucleocapsid protect the virus when it is outside the host cell. Genome. Figure 4. Schematic representation of the genome organization and functional domains of S protein for SARS-CoV and MERS-CoV. Coronaviruses contain a positive sense, single-stranded RNA genome. The genome size for coronaviruses ranges from approximately 27 to 34 kilobases. The genome size is one of the largest among RNA viruses. The genome has a 5-methylated cap and a 3-polyvinylated tail. The genome organization for a coronavirus is 5-letter UTR replicase transcriptase spike, S, envelope, E, membrane, M, nucleocapsid, N, 3-UTR poly, A, tail. The open reading frames 1A and 1B, which occupy the RST 2/3 of the genome, encode the replicase transcriptase polyprotein. The later reading frames encode the 4 major structural proteins: spike, envelope, membrane, and nucleocapsid. Interspersed between these reading frames are the reading frames for the accessory proteins. The number of accessory proteins and their function is unique depending on the specific coronavirus. Life cycle. Life cycle of coronavirus. 1 entry. Figure 5. The life cycle of a coronavirus. Infection begins when the viral spike S glycoprotein attaches to its complementary host cell receptor. After attachment, a protease of the host cell cleaves and activates the receptor-touched spike protein. Depending on the host cell protease available, cleavage and activation allows cell re-entry by endocytosis or direct fusion of the viral envelope with the host membrane. On entry into the host cell, the virus particle is uncoated, and its genome enters the cell cytoplasm. The coronavirus RNA genome has a 5-methylated cap and a 3-polydenylated tail, which allows the RNA to attach to the host cell's ribosome for translation. The host ribosome translates the initial overlapping open reading frame of the virus genome and forms a long polyprotein. The polyprotein has its own proteases which cleave the polyprotein into multiple non-structural proteins. Replication. A number of the non-structural proteins coalesce to form a multi-protein replicase transcriptase complex, RTC. The main replicase transcriptase protein is the RNA-dependent RNA polymerase, RDRP. It is directly involved in the replication and transcription of RNA from an RNA strand. The other non-structural proteins in the complex assist in the replication and transcription process. The hexoribonuclease non-structural protein, for instance, provides extradelity to replication by providing a proofreading function which the RNA-dependent RNA polymerase lacks. One of the main functions of the complex is to replicate the viral genome. RDRP directly mediates the synthesis of negative sense genomic RNA from the positive sense genomic RNA. This is followed by the replication of positive sense genomic RNA from the negative sense genomic RNA. The other important function of the complex is to transcribe the viral genome. RDRP directly mediates the synthesis of negative sense subgenomic RNA molecules from the positive sense genomic RNA. This is followed by the transcription of these negative sense subgenomic RNA molecules to their corresponding positive sense NAS. Release. The replicated positive sense genomic RNA becomes the genome of the progeny viruses. The RNAs are gene transcripts of the last third of the virus genome after the initial overlapping reading frame. These RNAs are translated by the host's ribosomes into the structural proteins and a number of accessory proteins. RNA translation occurs inside the endoplasmic reticulum. The viral structural proteins S, E, and M move along the secretory pathway into the Golgi intermediate compartment. There, the M proteins direct most protein-protein interactions required for assembly of viruses following its binding to the nucleocapsid. Progeny viruses are then released from the host cell by exocytosis through secretory vesicles. Coronavirus disease 2019. COVID-19. What is COVID-19?
Coronavirus disease 2019, COVID-19, is an infectious disease caused by severe acute respiratory syndrome coronavirus 2, SARS-CoV-2. The disease was first identified in 2019 in Wuhan, the capital of Hubei, China, and has since spread globally, resulting in the 2019-20 coronavirus pandemic. Common symptoms include fever, cough, and shortness of breath. Muscle pain, sputum production, diarrhea, and sore throat are less common. As of 23 March 2020, the rate of deaths per number of diagnosed cases is 4.4%. However, it ranges from 0.2% to 15%, according to age group and other health problems. The virus is typically spread from one person to another via respiratory droplets produced during coughing. It may also be spread from touching contaminated surfaces and then touching one's face. The virus can live on surfaces for 4 to 72 hours. Time from exposure to onset of symptoms is generally between 2 and 14 days, with an average of 3 days. Recommended measures to prevent infection include frequent hand washing, physical distancing, maintaining distance from others, and keeping hands away from the face. The use of masks is recommended for those who suspect they have the virus and their caregivers, but not for the general public, although simple cloth masks may be used by those who desire them. There is no vaccine or specific antiviral treatment for COVID-19. Management involves treatment of symptoms, supportive care, isolation, and experimental measures. The World Health Organization, WHO, declared the 2019-20 coronavirus outbreak a public health emergency of international concern, (PHEIC), on 30th January 2020 and a pandemic on 11th March 2020. Evidence of local transmission of the disease has been found in many countries across all 6 WHO regions. Symptoms. Symptoms of COVID-19. Cause. Cause of COVID-19. The disease is caused by the virus severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, previously referred to as the 2019 novel coronavirus, 2019-CoV. It is primarily spread between people via respiratory droplets from coughs and sneezes. Estimation of the rate of decay with a Bayesian regression model suggests that viruses may remain viable up to 18H on copper, 55H on cardboard, 90H on stainless steel, and over 100H on plastic. The lungs are the organs most affected by COVID-19 because the virus accesses host cells via the enzyme ACE2, which is most abundant in the type 2 alveolar cells of the lungs. The virus also affects gastrointestinal organs as ACE2 is abundantly expressed in the glandular cells of gastric, duodenal and rectal epithelium as well as endothelial cells and enterocytes of the small intestine. The origin is unknown, but by December 2019 the spread of infection was almost entirely driven by human-to-human transmission. The earliest known infection occurred on 17 November 2019 in Wuhan, China. Figure 7. Microscopy image showing SARS-CoV-2. The spikes on the outer edge of the virus particles resemble a crown, giving the disease its characteristic name. Figure 8. Schematic diagram of the coronavirus particle. S. Spike protein. M. Membrane protein. E. Envelope protein. N. Nucleocapsid protein. Structural proteins of coronavirus virion structure. Diagnosis. Diagnosis of COVID-19. Figure 9. CDC RT-PCR test kit for COVID-19. The WHO has published several testing protocols for the disease. The standard method of testing is real-time reverse transcription polymerase chain reaction, RT-PCR. The test can be done on respiratory samples obtained by various methods, including a nasopharyngeal swab or sputum sample. Results are generally available within a few hours to 2 days. Blood tests can be used, but these require 2 blood samples taken 2 weeks apart, and the results have little immediate value. Chinese scientists were able to isolate a strain of the coronavirus and published the genetic sequence so that laboratories across the world could independently develop polymerase chain reaction (PCR) tests to detect infection by the virus. As of 19 March 2020, there were no antibody tests, though efforts to develop them are ongoing. The FDA approved the RST point-of-care test on 21 March 2020 for use at the end of that month. Diagnostic guidelines released by Zhongnan Hospital of Wuhan University suggested methods for detecting infections based upon clinical features and epidemiological risk. These involved identifying people who had at least 2 of the following symptoms in addition to a history of travel to Wuhan or contact with other infected people: fever, imaging features of pneumonia, normal or reduced white blood cell count, or reduced lymphocyte count. A study published by a team at the Tongji Hospital in Wuhan on 26 February 2020 showed that a chest CT scan for COVID-19 has greater sensitivity, 98%, than the polymerase chain reaction, 71%. Figure 10. Typical CT imaging findings. Figure 11. CT imaging of rapid progression stage. Prevention. Prevention of COVID-19. Figure 11. An illustration of the effect of spreading out infections over a long period of time, known as arcing the curve. Decreasing peaks allows healthcare services to better manage the same volume of patients. Figure 12. Alternatives to arcing the curve. Figure 13. Prevention efforts are multiplicative, with effects far beyond that of a single spread. Each avoided case leads to more avoided cases down the line, which in turn can stop the outbreak in its tracks. Figure 14. Regulatory action whilst 2019-20 coronavirus pandemic in shops, gap of at least 1.5 m between people. Management of COVID-19. Figure 14. 4 steps to putting on personal protective equipment. Prognosis of COVID-19. Figure 15. The severity of diagnosed COVID-19 cases in China. Figure 16. Case fatality rates by age group in China. Data through 11th February 2020. Figure 17. Case fatality rate in China depending on other health problems. Data through 11th February 2020. The disease may take a mild course with few or no symptoms, resembling other common upper respiratory diseases such as the common cold. Children of all ages are susceptible to the disease, but are likely to have milder symptoms and a much lower chance of severe disease than adults. In those younger than 50 years, the risk of death is less than 0.5%, while in those older than 70 it is more than 8%. Pregnant women are at particular risk for severe infection. Complications associated with COVID-19 include sepsis, abnormal clotting, and damage to the heart, kidneys, and liver. Many of those who die of COVID-19 have pre-existing conditions, including hypertension, diabetes mellitus, and cardiovascular disease. Epidemiology. Epidemiology of COVID-19. Figure 18. Total confirmed cases over time. Figure 19. Total deaths over time. Figure 20. Total confirmed cases of COVID-19 per million people, 20th March 2020. Figure 21. Total confirmed deaths due to COVID-19 per million people, 24th March 2020. Terminology. Terminology of COVID-19. The World Health Organization announced on 11th February 2020 that COVID-19 would be the official name of the disease. World Health Organization Chief Tedros Adhanom Ghebreyesus said, CO stands for Corona, 6 for virus, and D for disease, while 19 was for the year, as the outbreak was RST identified on 30th December 2019. Tedros said the name had been chosen to avoid references to a specific geographical location, i.e. China, animal species, or group of people in line with international recommendations for naming aimed at preventing stigmatization. While the disease is named COVID-19, the virus that causes it is named Severe Acute Respiratory Syndrome Coronavirus 2 or SARS-CoV-2. The virus was initially referred to as the 2019 novel coronavirus or 2019 GOV. Research. Research of COVID-19. Vaccine. There is no available vaccine, but research into developing a vaccine has been undertaken by various agencies. Previous work on SARS-CoV is being utilized because SARS-CoV-2 and SARS-CoV both use the ACE2 receptor to enter human cells. There are 3 vaccination strategies being investigated. On 16th March 2020, the RST clinical trial of a vaccine started with 4 volunteers in Seattle. The vaccine contains a harmless genetic code copied from the virus that causes the disease. One difficulty with vaccine development is that older people, who are more vulnerable to the disease, are often poorly vaccinated due to age-related degradation of the thymus. Antivirals. Several existing antiviral medications are being looked at to treat COVID-19 and some are moving into clinical trials. In March 2020, WHO has launched a multi-country trail involving 10 countries called Solidarity, in response to COVID-19 pandemic. Remdesivir, chloroquine and hydroxychloroquine, ritonavir lopinavir and ritonavir lopinavir combined with interferon beta are the experimental treatments currently being researched under Solidarity trial. On 17th March, the Italian pharmaceutical agency included chloroquine and hydroxychloroquine in the list of drugs with positive preliminary results for treatment of COVID-19. Korean and Chinese health authorities recommend the use of chloroquine. However, the Wuhan Institute of Virology, while recommending a daily dose of 1 gram, notes that twice that dose is highly dangerous and could be lethal. As of 20th March 2020, The treatment has not yet been approved by the US Food and Drug Administration. Anti-cytokine storm. Cytokine storm, a life-threatening medical condition, can be a complication in the later stages of severe COVID-19. There is evidence that hydroxychloroquine has anti-cytokine storm properties. Tocilizumab has been included in treatment guidelines by China's National Health Commission after a small study was completed. It is undergoing a phase 2 non-randomized test at the national level in Italy after showing positive results in people with severe disease. Combined with a serum ferritin blood test to identify cytokine storms, it is meant to counter such developments, which are thought to be the cause of death in some affected people. The interleukin-6 receptor antagonist was approved by the FDA for treatment against cytokine release syndrome induced by a different cause, CAR cell therapy, in 2017. The Feinstein Institute of Northwell Health announced in March a study on a human antibody that may prevent the activity of IL-6. Passive antibody therapy. Transferring blood donations containing antibodies produced by the immune systems of those who have recovered from COVID-19 to people who need them is being investigated as a non-vaccine method of immunization. This strategy was tried for SARS. Viral neutralization is the anticipated mechanism of action by which passive antibody therapy can mediate defense against SARS-CoV-2. Other mechanisms, such as antibody-dependent cellular cytotoxicity and/or phagocytosis, may however be possible. Other forms of passive antibody therapy, for example, using manufactured monoclonal antibodies, are in development. Production of convalescent serum, which consists of the liquid portion of the blood from recovered patients and contains antibodies specific to this virus, could be increased for quicker deployment. Thanks for watching our videos. Please subscribe and share these videos. Download ebooks, Kindle, for just 50 rupees at Amazon. Download ebooks, PDF, for just 25 rupees at PayPal. Note: links are given below in videos descriptions. Go Corona, Corona go. Go Corona, Corona go. Go Corona, Corona go.